When people think of life in the seas, it is often the majestic that comes to mind, such as whales, sharks, rays, and coral reefs, or even the fish that feeds us for our own sustenance. Rarely do we think of plankton, the tiny organisms found across the world's oceans, which are responsible for most of the air that we breathe and the food that we eat. This is why we're here, because by standing up for the smallest organisms in the open ocean, we're standing up for all animals in the seas. Good morning, ladies and gentlemen. Welcome to this year's fourth Philippine Planktology lecture series with the theme Unraveling the Secret of Oceanic Enigmas and Rare Planktonic Life Forms. To formally open our three-part lecture series this year, let us all welcome the current administrator of the Philippine Planktology, Dr. Mark Louis Lopez, for his opening remarks.
Thank you, Darlene. Hello, everyone. Good morning, and welcome to the fourth Philippine Planktology lecture series. As mentioned by Darlene, this year's theme for the lecture series, we have Unraveling the Secrets of Oceanic Enigmas and Rare Planktonic Life Forms. So, just to acknowledge that this event is held in collaboration with the UP Visayas Institute of Marine Fisheries and Oceanology, our hosting institution, the Plankton Society of the Philippines, and Plankton Niche Laboratory headed by Dr. Christy Acabado in UP Visayas. So, 4 years ago, during the pandemic, we started this initiative of organizing online lecture series to hopefully raise awareness and share the latest updates in the field of planktonic systematics, or plankton systematics and ecology. And this somehow diversified to related taxa, not only to planktonic communities. And you'll see later with today's talk how it kind of diversified with parasitic organisms, but still on the same taxa, which are mostly crustaceans. So throughout or over the 4 years, it has been a pleasure to witness the growing interest and participation from different audience, mainly coming from or mainly consist of university undergraduate students and grad students, university researchers, and government scientists. So for this year's lecture series, we hope that you enjoy the different talks and also continue to nurture your curiosity about planktonic communities and related taxa. So thank you for joining us and supporting the advancement of plankton ecology and ecology research in the Philippines and Southeast Asian region. So thanks, everyone.
Thank you, Dr. Louis, for your opening remarks. So our program for today is roughly 1 hour and 30 minutes. And before we proceed to our program, may we remind everyone of a few house rules in order for us to have a more orderly virtual lecture. First, participants are requested to turn off their cameras and keep their microphones on mute when not speaking. Second, please hold your questions until the end of the presentation because questions will be entertained during the open forum. And then to ask a question during the open forum, kindly raise your hand and wait for the moderator to call you in. And then you may also type in your questions in the QA box, and please do not spam the chat box. And lastly, kindly accomplish the evaluation form on the link provided in the chat box to claim your e-certificate for this activity. Now, I'm sure everyone is excited to meet our speaker for today. So without further ado, may I call in the current director of the Institute of Marine Fisheries and Oceanology and also currently the Vice President of the Plankton Society of the Philippines, Dr. Christy S. Acabado, to introduce our speaker this morning. Ma'am.
Yeah. Good morning, everyone. Thank you for joining us in our webinar today. Our speaker, I met her on a Facebook page when I found a rare louse from a dolphin that we tried to rescue a few months ago. So you see, we find the rarest scientists in the most unexpected places. Our speaker for today is from the Laboratorio de Diversidad Genómica from University of Sao Paulo, Brazil. She is a Brazilian systematic biologist with interest in ecology and evolution of associations between crustaceans and marine vertebrates. She is passionate about the ocean and islands, working on the phylogeography and connectivity of paracarp crustaceans from remote islands. Currently, she is a postdoctoral researcher at the University of Sao Paulo, Brazil, and involved in research projects mostly related to taxonomy, phylogenetics, biogeography, and molecular ecology of crabs and amphipods. Ladies and gentlemen, let us welcome our speaker, Dr. Tammy Iwasa, Arai.
Hi, Christy. Thank you for the introduction and thank you for the invitation. Can I share my presentation? Yeah? Guys, so just let me know if it's showing for you and if you can hear me, please.
Yeah, it's very clear.
Pardon?
Yeah, it's very clear, the presentation and your volume, audio.
Perfect. Okay. So, well, thank you for being here today. I'm very happy to share some things that I know about whale lice, and I hope you enjoy it. So, I'm Tammy. I'm from the University of São Paulo in Brazil. So now it's like 10 p.m. here. So good evening for you. So let's start. So my presentation, well, hang on, it's not working. Okay, so the presentation is organized into one part where I'm gonna talk a little bit about the whale lice biology, so classification and about morphology and some of the most common questions that I receive. And then I'm gonna talk a little bit about the hosts and its interactions, then about the recent findings that we are doing now and some of my work and some other colleagues that are working with whale lice, and finally, the current and future studies. So first of all, who are they? Who are the whale lice? Actually, they are not real lice. They are not insects. They are, in fact, crustaceans from the order Amphipoda, and all of the whale lice belong to the family Siamidae. So all the whale lice are Siamids, and all Siamids are whale lice. Like there's only whale lice inside this family, and this family it's subdivided into two subfamilies, Siaminae and Isosiaminae. And if we look here into the general body shape of amphipods, we can see that they kind of look like the same in this laterally flattened body. So this compression of lateral compression is very common in amphipods, but not for the whale lice. So I don't know if you can see my arrow here, or my blue spot here, but the whale lice, they are in fact dorsal-ventrally flattened, and because of this, they put the earlier researchers put them before into another order, the isopods, where we can have like the wood louse, for instance. And but because of this general morphology, they put them into isopoda. And when we have a deeper look into their morphology, we can see that they are in fact amphipods, and its sister, the sister clade of whale louse is the skeleton shrimp, which is this one here. So we can see that when we have a first look, they look like very different from each other, but they are actually very similar. So here I have a picture of an gammarid amphipod. As some of the people here know, they are like the body is separated into head, the perion, the pleon, and the urosome. So they are in different colors here. And all the legs are in the perion. And then we have like the pleopods and all the other appendices. But for the whale lice, they are actually very they have reduced body parts because they need to attach to the whale or to the dolphin. So they have very strong claws in all the legs, and they have a large perion, but a very, very reduced pleon. So here we can see like a detailed morphology where we can have the antennule, or the first antenna, which is the largest one, and then the smallest one is the antenna. We have gills. So in these segments of the perion, they don't have legs. They just have these gills. And for instance, in isopods, the gills are on the pleon, not in the perion. So that's another difference that makes them amphipods. So we have these long gills and the lack of periopods, the legs. And they also have this accessory gill, which is a very important feature, like a character for the identification of each species of whale louse. They also have two pairs of gnathopods, like all the other amphipods. The females have a marsupial, which are composed of 4 plates of the osteoid in the perionids 3 and 4, and they form a marsupial where they can have their eggs, and the eggs are hatched, and then the juveniles go from the marsupial to feed on the whale and then go back to this marsupial like a baby kangaroo. And we also have the genital valves on females and a very, very reduced pleon here, as you can see. Well, and if we see the differences between males and females, there is sexual dimorphism. Here I put some characters from the same species. So we have the accessory gills of a male of Siamus bupis, which is bilobed here. And on the females, it's more like rounded shaped. So the accessory gills are very important for the identification of the species, but not only for the species, but also for the sex of the species. We can also have in females the genital pore on the perionid 5 and the marsupial on perionids 3 and 4. And these are the plates that I told you before. So we have 4 plates making this marsupial, and the male has 2 penis. And this is, I think, the most common question that I receive, like, how do we sample them? Because they are actually in whales, so like on the top of the whales. So how do we sample them? So this is actually what everybody thinks I do, that I'm always swimming with whales and doing this research in a beautiful environment. This is actually what my friends think I do, which is collecting from a stand-up paddle and just trying to grab them from the top of the whale. But in fact, what I really do is to wait for a dead whale to beach and then strand on the beach. And it's actually a very smelly and not really nice work, but indeed we have fruitful results. So yeah, we don't collect them from live whales unless they strand on the beach alive. But most of the samples are from the time when whaling was permitted. So most of the huntings gave us over 25% of the species that were described. So between the 17th to 1950s, most of the species of whale lice were described. And nowadays, we can only find these samples on fresh beached carcasses. So this means that most of the samples are from coastal and abundant species, such as the humpback whale or the right whale or the gray whale, for instance, or some other small cetaceans, such as the bottlenose dolphin and other coastal species. And this means that some of the samples that we have, especially from beaked whales that are pelagic or other species that are not that abundant, we only have samples in museums, in collections. And the second most common question that I receive is, how many lice do a whale have? So, well, actually, I don't know, it depends on the whale, but here we have one batch that I received last year from the last season of whales here in Brazil. So this batch is around like 30 centimeters long. And I asked in my lab, how many lice do we have here? And they guessed like from I don't know. Can you guess on a chat? Then we can have a look later. But if you want, just put some number in there. I can show you here that all these white patches are actually dead skin or blubber, but all of like the rest of the things inside are whale lice. So I don't know how many actually. But in my lab, they guessed from 300 to over 12,000, no, 1,000, no, 12,000, yeah, 12,000. So yeah, we have like different guesses in there. But in 1975, Leung already estimated the abundance of of gray whales in over 100,000 Siamus scamoni, which is the species of whale lice, in one gray whale host. So we know that over 100,000 whale lice can live on whale. And it was very funny because last week I received this video, and it's from Brazil now, and it was like passing like with a very romantic soundtrack and written like the pink whale, and all the comments in the social media was like, how beautiful is nature, how perfect is nature. And actually it's only pink because of the whale lice, because it's only like all covered by whale lice. And this means that this whale is not healthy. Whale lice can co-evolve with whales, so they are not harmful if they are in very small amounts. But if we see this large amount of whale lice, it means that something happened to this whale to be totally covered by whale lice. So yeah, well, indeed, nature is perfect and nature is beautiful, but yeah, just because I like parasites, maybe. Some other curiosities of the whale lice is that they have direct development, and by that I mean, I'm sorry guys, but they don't have a larval phase, and I'm sorry to disappoint you, all the planktologists, but they don't live on the plankton. I think you'll never see one whale louse on the plankton, but please, if you do, just contact me because it would be very nice to know. And about some fossil records, we know that there's Sirenoceamus retine, which was one species described for manatees and dugongs. And that's why it's called Sirenoceamus, because of Sirenia. But it was found on a piece of flesh that was floating in the ocean, and they thought it was from a dugong, but indeed nowadays it's not accepted species. But in fact, there is a fossil record of a Siamid from the Holocene, so very recent. And they also have host specificity, and this means that for Mysticeti or the baleen whales, they have host specificity, but not for odontoceti, which are the dolphins and toothed whales. So they are generalist in toothed whales, but host specific in mysticeti. So what does this mean, right? That means that one species of whale louse can be in only one species of host for the baleen whales, but for the dolphins and toothed whales, one species can be found in lots of host species. And Siamus, which is the largest genus, is found in both baleen whales and dolphins. And Balenosiamus, which is another genus, is only found on species like the rorquals, like the whales that look like submarines, such as the blue whale, the minke whale, and the fin whale. So it's only found on them. And several other genera are found in dolphins and toothed whales, such as Isosiamus, Neosiamus, Orcinosiamus, Platisiamus, Scutosiamus, and Cinciamus. And when I told you that one whale louse can be present in only one host species, I mean that, for instance, for the humpback whale, which is the most abundant species here in Brazil, so they only host one species of whale lice, which is Siamus bupis, but other species of whales, such as the gray whale, can host 3 species of whale lice. For instance, Siamus kessleri, Seti, and Scamoni. So they are very species specific, and the right whale can also host 3 species: the Siamus ovalis and Siamus gracilis, and the Siamus erraticus. So not only they have species-specific interactions, but they also have isolated niches for each species. So, for instance, Siamus ovalis and gracilis, which are these 2 species here, they can be found on the head of the right whale. And on the head of the right whale, I don't know if you saw some pictures of right whales before, but they are like all black. But on the head, they have some things that look like rocks on the top of the head and here. And these patches are rough patches of rough skin called callosities. And these callosities are actually black, but because they are all covered by whale lice, they turn into like white. And then since each host individual have a different shape of this callosity, because of the whale lice on the top, they can see from like a surveys, aerial surveys, which individual is which. So they can photo identify each whale based on actually the whale lice that they have. So these 2 species occur on the top of the head of the whale, but another one is found on wounds and on the urogenital region, which is Siamus erraticus. And actually, if we think about the transmission of the whale louse, they cannot swim. They don't have a larval phase, and they cannot swim as adults. So they can only be transmitted from one whale to another if 2 whales get in touch. And the first time that a baby whale have an infestation is when the calf is born, because they have in this urogenital region the whale lice there, and then when it gives birth to the baby whale, these ectoparasites actually go to the head of the whale's calf. And then they started having this species here, Siamus erraticus, on the head. But during time, they change to other species. So this Siamus erraticus, it's more like orange, and they say they can see these orange patches on the top of the head of the baby whale. But then after some time passes, some weeks, these patches change color, and this means that these Siamus erraticus move from the head to the wounds or to the urogenital region of the calf. Okay. And this means that the right whales have these 3 species. So right whales already have 3 species of whale lice. It's the most diverse environment for a whale lice to be. But there was one time 10 years ago when a baby right whale, stranded on the beach alive, and then they put it to put it back to the water. It swam like the baby swam for 1 day and then stranded again, already dead. And I received these samples when I was in my master's, so I was just identifying some whale lice. And when I saw on the label, it was written right whale. And the whale lice that I identified, they all were another species, Siamus bupis, which is only found on humpback whales. So I was thinking, why this calf had only Siamus bupis? Because like it's a species that should be in another species of whale. So we wrote a note saying that this was an adoption of a baby whale from another species, and like the reviewers thought like maybe this is too exaggerated, so we changed the title and we put like the possible interaction of a southern right whale with a humpback whale. So yeah, we thought maybe we reported the first interaction between humpback whales and southern right whales, and yeah, that was it. But last year, or actually 2 years ago, I saw this. I saw in like an Australian newspaper this article that was a young humpback whale photographed with southern right whales. And then there was this picture from a drone where we can see an adult right whale here. And you can also see the white patches on the head, on the top of the head. So this is the right whale swimming with a humpback whale, a calf of humpback whale. So this interaction apparently is more common than we expected. Like actually we saw the opposite, baby right whale with an adult of humpback whales, and they saw the opposite. But it means that these interactions are not that rare. And last year we also found this bottlenose dolphin with also the same species, Siamus bupis. And bottlenose dolphins are very playful. They are always interacting with other species of cetaceans. So we thought, oh, okay, maybe it interacted with humpback whales, but we've never seen this occurrence, like this report of Siamus bupis in dolphins before. And I was thinking like, oh, okay, so maybe they are not that host specific, but all these records were from dead animals. So something happened to these hosts, like these individual hosts. And that's why probably the species doesn't continue going on, because all these hosts were ill or injured and they happen to die. And then they cannot be generalized because these interactions are very punctual. So here we used the same species, Siamus bupis, to investigate the genetic population of these whale lice in different populations of humpback whales in the world. So humpback whales spend half of the year in feeding grounds on the poles. So talking now about the southern hemisphere, in the southern hemisphere, they spend the summer in Antarctica and they migrate to the tropics during winter, where they can breed and have their offspring. So this every year they migrate to the poles, to the tropics, and not only here from the Antarctica to the Brazilian part, but also to other regions of the world. So we sampled some whale lice, not only from Brazil, but also from Namibia. Namibia is actually here, it's just because it would be very close to each other. But so some from South America, from Africa, and from Australia. And we also used some sequences of CO1 from the northern hemisphere. And we tried to see if they were isolated, because we expected that since they don't have a larval stage and they don't swim, they only spend their life in one whale. So we expected very, very strong isolation of these populations. But instead, we saw that the populations from South America and Australia were much more alike than comparing the South America to Africa, and both of them were collected in the Atlantic. So it means that maybe they are changing this whale lice in the Antarctic region, where they all share this feeding ground, or maybe some of these whales, they crossed and changed populations during time. So this was actually not the first genetic study on whale lice. Kalizewska published the first study in 2005, where they studied the three species that I already told you about. So Siamus ovalis, gracilis, and erraticus from right whales. And the right whales, they not only have three species of whale lice, but they are actually three species of right whales as well. So there's one species that is just distributed in the North Atlantic, another one that is on the southern part, the southern hemisphere, and another one which is only present in this part of the Pacific. So they used samples from all these three species of right whales and the three species of whale lice. And they found that each whale lice was actually isolated into these three species of whales. So here we have Siamus erraticus. And it's divided into Southern Hemisphere and North Atlantic. The same happens for Siamus gracilis and Siamus ovalis here. So there is one clade from the North Atlantic and another one from the Southern Ocean, which is shared with the Northern Pacific. And another work was done by Callahan in 2008. It was a master thesis that was not published, and where he investigated three species that occur on the gray whale, so Siamus escamoni, Seti, and Kessleri. And he saw that the three species that occur on the gray whale, they are not sibling species. So that means that these colonizations of a host happened multiple times. Not only one colonization and then the speciation, but several colonizations. So these are the three species of hosts that were previously studied using whale lice, and some of the ecological work that has been done for whale lice, like the most important ones were from Victoria Roundtree in the 1990s, where she saw this whale lice shift from one species to other on the head of the right whale. So she saw that it was changing places with other colonizations. She also studied the proportion of gravid and non-gravid females in one host, and also the size of the whale lice. So it was like a very important work in the beginning. And Natalia Freja Fernandez also did some work on another species, which is Cincia musicus. And the difference is that Cincia musicus actually occurs on dolphins. So it's much more difficult to have these samples because dolphins have a very small abundance of whale lice. So, for instance, if in baleen whales we have over 100,000 whale lice, in dolphins they have numbers like of 3 or 1 or very small amounts of whale lice in one host. For instance, there was one species that I described from one big whale, and it was from a mass stranding. So 7 whales stranded all together in New Caledonia, and I received only 1 whale louse. And I was like, okay, but why there's only 1 whale louse if they have like 7 big whales there? And in fact, there was only just 1 whale louse in all the 7 hosts. So I don't know, we don't know very well how they can still like live on these dolphins, but they can perpetuate apparently. So here in this work, Natalia saw the presence on the blowhole and the corner of the mouth and the eyes. So also in dolphins, they have some prevalence in different sites of the dolphin as well. And about the systematics, this was the first study made by Henny in 1999 in a master thesis as well. And 1 year later, Margolis proposed another phylogeny for whale lice, where he used numerical analysis. And in 2018, I guess, we published this work using the whale lice that we had. And all in these 3 works, we can see that there are 2 clades. So one in which they have all the genera found on dolphins and toothed whales, and another clade which is comprised of Siamos and Belenosiamos. So we called that the 2 subfamilies, Isosiamine and Siamine here. And about the current and future studies, during my postdoc here now, we are trying to investigate the genomic adaptation to epibiosis. So we are sequencing the first genome of a whale louse. We are also trying to do some phylogenomics on whale lice using UCs, which are ultra-conserved elements that are present on the genome. So this is actually the currently accepted phylogeny, and it's all based on morphology. So now we want to sequence more data to try to include and try to understand better this phylogenomics, and also try to do some co-phylogenetic analysis. So using both the phylogenomics of whale lice and try to correlate and to compare with the cetaceans phylogeny. This is part of my work now, but there are some other colleagues that are working with whale lice right now. So Sofia Ten from the Universidad of Valencia, she's working with Cincia musicus population genomics. She's doing also UCs to try to understand the population structure of Cincia musicus. And Pedro Pereira, here from Universidade de São Paulo, he's an undergrad student and he just started working with Siamos Bupis population biology. So we are trying to do some works like the Victoria Hound tree for the ovalis and right whale lice. We are trying to do for humpback whales now. So yeah, that's it. I would like to thank you all for watching my presentation. I'm happy to respond questions now. And these are all the collaborators that gave us samples and support our work during this time. Thank you.
Thank you so much, Dr. Tammy, for that very interesting lecture on whale lice. We are very interested in your work. It's very interesting. And thank you for sharing some of your current and future plans on genomics and systematics of whale lice. And I'm sure everyone here has some questions about the talk. So may I call in Dr. Christy Acabado as our moderator for the open forum.
Yes. Oh, yes. Thank you very much, Dr. Tammy. We received several emoji reactions in most of your slides, and I believe most of our participants here are now getting more interested in like finding stranded whales, but we hope we don't find a lot. But actually, in the Philippines, we especially here in our region, we find a lot of strandings. Like in the past 6 months, we already have four, at least four in our city. So but now I don't think we have an expert on Laos here in the country. So if because we have a lot of students here, maybe the first question is, if, for example, some of our students come across stranded whale or dolphin, how are they supposed to spot a very, very tiny louse on a dolphin or a whale, and how should they collect them so that maybe we can start also doing something on this?
Yeah, it would be very nice if you have like a start a research on whale lice there. Maybe you could start like collecting the samples like manually or with a Twizzle and store on ethanol, because ethanol from 70 to 90%. Formaldehyde, it's not like used anymore. And yeah, you could store them on like just a regular freezer and try to look to that characteristics that I mentioned, such as the accessory gills and the number of spines on the perionates. Some of that are important features for identification. There are also two identification keys for whale lice. So maybe they could use the one from Leung. I think it's 1970 something, but it's very useful. There are like not all the species are there because other species were described later on, but it's very useful. And I think we have one identification key in our paper as well, but maybe if only for genus. I don't know. I don't remember actually. But yeah, you could start with that. And I'm very happy to help if you need any assistance.
Yes, so I guess a lot of people are not now interested in doing collaborations with you. So we have here questions from the Q&A box. I guess these are from students in the audience. So one is, Thank you, Dr. Tammy, for sharing your expertise. I would like to ask about the number of lice found in whales. Is this counts per individual, or are there methods to standardize the counts, perhaps the density?
Okay, so these are like the we don't know the regular number of a healthy whale. This is one question that we want to respond, but we don't know. But we know that when it's all covered by whale lice, it's like
Not
it's one
healthy.
in 1,000. I don't know, and
Oh.
yeah, it's not very common. And most of the time, the whale is very ill or very thin. You can see that something happened. But these estimates are, there's not an average, there is not a mean. So yeah, I don't know how many there are
Mhm.
in one healthy whale. Yeah. We know
Okay,
that
thank
these are like blooming ones are not healthy. And we know that it's over 100,000, but I think nobody counted before.
you. Okay, yes, thank you for that answer. I have another question here from the Q&A box. Is it possible to use the presence of whale parasites to map out the locations where whales travel around the world?
Whoa, yeah, well, I mean, I don't know if we can say that this individual traveled around the world, but we can say that one individual during the time traveled because we know that, for instance, in for right whales, there are three species of right whales. Uh, they still are the same species, so they are isolated, but they still share some features. And for that, that means that one whale at least must cross from one ocean to another to interchange this whale lice. So yeah, I told one time in one interview that whale lice are more useful to try to understand the migration patterns, because when we put a tag on a whale, we can only track one whale, and when we study the whale lice, we can have a deeper look on the interactions between these individuals. So we know that sometimes they interacted with other populations because of this whale lice interchange, but we don't know which individual crossed, for instance.
Mhm. Okay, yes,
I
thank
don't know
you.
if I
So,
responded, but
yeah, of course. In the audience, we actually have one 7-year-old here, and she's a bit sad about the dead baby whales. And she has a lot of questions, but this is one of them. Is there a way to know? Sorry. Are there whale lice inside dolphins or whales, like for example in the mouth?
okay, no, they only live on the outside part of the whale, so they eat actually, oh, I forgot to tell them, but they actually eat the dead skin, so they're not that harmful. They only eat like the first part of the skin. But when a whale have some wounds, for instance, some wounds or the blowhole, they can go into that parts, but not like live inside the whale, no.
Oh, okay. So interesting. I I know that there are copepods, parasitic copepods also on whales and dolphins. So have you ever come across them, or do you know anybody working on them?
Okay, so I've seen like once only. They are not very common here on the whales that I found. But yeah,
Mhm.
it's Penella. It's a big copepod, but there are also copepods living on the baleen. So the baleen is the thing that they have instead of the teeth.
Mhm.
So there are some species that only lives on these baleen plates. But this one
Mhm.
I have never seen before.
Okay. So we have two people raising their hands from the audience. Can I call on Edward Joshua Mendoza? Sir Junie, can we allow him to turn on his microphone? Oh, and we have another one, Kim Nunez. Hi, Kim. Oh, we lost them. Or maybe they just pressed it accidentally. Anyway, so we have another question
Hello,
here.
Ate
Oh
Christy. Ate Christy.
yeah, oh yeah. So Kim is here. Hi, Kim.
Actually, it's Lors. Using
Oh, hi,
Kim's
Lors.
laptop.
Hi, Lorely.
Yeah. I have I don't know if I missed this part, but I think you already mentioned earlier, the speaker already mentioned earlier that the whale lice incur some wounds in the whales. But other than that, do they affect other physiological functions of the whales or reproductive behavior or reproduction of the whale of the host? Other than the physical effect, what other impacts does the Siamus parasite incur to the host? Do they affect the behavior or the reproduction, for example, or even the physiological aspect of the host?
Okay, thank you for the question. This is actually a very interesting question. We don't know anything about the what the whale lice can cause on whales. We don't know if they have some viruses that they can transmit, for instance, some diseases. We don't know. We are trying to do some things now, maybe a verum, but for now, there are no research about anything associated to behavior or anything else on whale lice. There's only one hypothesis, that is that whales sometimes jump outside the water to remove these ectoparasites, but that's only this that people hypothesized and discussed before. Apart from that, we don't know anything. Actually, well, nobody works with whale lice besides me and Sophia.
Yes.
And Dr. Leonard in Germany, I guess. But yeah, there are a few specialists. So yeah, it's always nice to have some people interested in doing some other works.
Yeah, I
Yeah.
asked the question because I also work on isopod parasites. So in crustaceans, usually the parasite affects the reproduction, so they prevents the host to reproduce, for example. So it would be really interesting also to look at such field, or yeah, to answer such question for this type of parasite. Thank you so
Yeah.
much for answering the question.
Thank you,
Thank
Lors.
you.
Thank you. Thank you for your question. Yeah, so I guess this webinar is really a good venue to introduce different kinds of fields in science. I hope from here we were able to inspire students to work on something very rare. So thank you. And then we have another question here from the Q&A box, maybe from one of our students here on the participants. So just a curious question. Are barnacles considered to be parasitic in whales, or are they just, free riders?
Okay, so barnacles are, well, they don't feed on whales and dolphins, but they are very heavy. So they are not considered parasites themselves, but when they are like in large amounts, they can make the host swim slowly, and this can change all the behavior of the host. So yeah, it's a difficult question. I would say no, but I also would say maybe. It depends.
Yeah, correct. Okay, so I have another question, maybe the last question. Yeah, so we do not have
I think
questions here
there's a
anymore.
question, Christy. I think
Yeah.
there's a question on the chat.
Oh, on the chat. Okay. Yes. So, hello, Dr. Tammy. Can we consider these lice as bioindicators of marine environment where these whales or cetaceans are?
I think they can be a bioindicator of the individual itself, but not for the environment. It depends on the prevalence. So if we start to see that here, for instance, in Brazil, we have several whales with a lot of whale lice, but not in other parts of the world, then maybe. But for now, we don't have anything like this. But it's always nice to to observe if this pattern is changing.
Yeah, so I guess for the participants, we can help out our the few specialists in the world. So by when we see strandings or stranded dolphins and whales, maybe we can try to check and observe their whale lice, and maybe we can contribute to science this way. Okay, so um, uh, I have another question here. So maybe the last. So when the whales or dolphins move from the poles to the tropics, do lice stay with them without dying? So this is about maybe the resilience of these lice to temperature change.
Yeah, so yeah, we don't know that as well. There are a few words on whale lice, as you can see. I could say like most of them in less than 1 hour. We don't know if they change, but we were trying to think of an experiment to see the thermal tolerance and also the pressure tolerance of them, because some species migrate from the poles to the tropics, but some species dive very deep. So we wanted to analyze that. But for that, we would need them alive. And it's very difficult to do experiments with whale owls without the whale.
Yeah.
Because they feed on a whale, they live on a whale, and it's very hard to try to cultivate, like create them in the lab. But yeah, it's a very interesting question.
Yeah, so there are a lot of questions that we cannot really answer because we don't really dive with the whales. Yes. So thank you very much for answering all of those questions, and thank you for everyone who contributed questions. Oh, but we still have one more. Maybe we should entertain this one. Yeah. So you mentioned dolphin have lesser parasites compared to whales. Is it attributed to its swimming behavior because dolphins are faster swimmers than whales?
Yeah, that's it. So, well, we don't have like an answer. Like, we cannot try to prove that, but scientists think that it's because of the speed of dolphins and because they are more like they are smooth, they don't have anything to attach on. So for instance, whales have also barnacles that are on the top, and then it's easier for us whale owls to cling on, to grasp on. So I think that's why it's easier for a whale louse to live on the whale, not in dolphins. So because of the speed, because of the body shape of the whale, it's also easy.
Yeah, flatter. Yes. So I remember when I took the whale louse, the louse from the dolphin, it was really difficult. It was really clinging on to the skin. So I didn't
Yeah.
have forceps during that time, so I just took it with my fingers. So anyway, so we have it now. So I think the participants are just getting warm. We have another question here. uh, uh, does this parasitic lice dies when the parasitic lice die? Oh, no, sorry. So when the host dies, does the parasitic lice die also, or can it still survive?
Well, it can still survive for a while. I mean, when the flesh gets like very, I don't know, like starts to decompose, they they can detach, but they will die eventually because they cannot swim. So they will just like sink to the bottom. Or just only if another whale passes by. But for instance, in strandings, when we have a dead whale and the researchers are collecting samples, for instance, the whale lice can start wall crawling on the researchers. I think they
Oh,
sense some thermal thing, I don't know, some temperature, but I don't know why, but they start clinging on us. So, but since we are not going to the ocean, they will die eventually.
yes. Okay. So that's quite interesting. Oh, we have one more question here. So, do you usually find male and female louse in one host?
Not always, yeah. So in this paper that I showed you before from Fraga Fernandez, they didn't find males and females in one host. And also when I described that species based on one specimen, it was only the male. We never found the female. We don't know how the female is. Uh, so yeah, it's a very deep discussion because we don't know how whale lice from dolphins. Uh, I don't know how they can keep living. Yeah, we don't know that
And
because they are
reproducing.
very small amounts.
Yes, right. Okay, so we still have a lot of questions to answer about the whale louse. So I guess it's time for a lot of our students here to, consider going into this field so we can add into this field full of experts. Okay, so thank you very much. I think we should move on with the program. Thank you very much, Dr. Tammy. I would like to pass the virtual stage to our host, Darlene. Thank you.
Thank
Thank you
you.
so much, Dr. Tammy and Dr. Acabado, for the that interesting exchange of ideas. So at this juncture, we would like to award the certificate of appreciation to our speaker. So I will read the citation. This certificate is awarded to Dr. Tammy Iwasa Arai in recognition of her outstanding contribution as keynote speaker of the presentation entitled Whales Cleanup Crew, Ecology and Evolution of Whale Lice at the fourth lecture series on Philippine Planktology on July 15, 2024. Signed, Dr. Christy Acabado, the Director of Info and also the Vice President of the Plankton Society of the Philippines. Also signed by Dr. Mark Louis Lopez, the current Administrator of Philippine Planktology. So thank you so much, Dr. Tammy. And may I request everyone to please open their camera so we can have a photo opportunity with our speakers and also our participants for today. Okay. Okay, I'll take the photo. Wait a minute. Okay. 1, 2, 3, smile. Okay. So thank you so much, Dr. Tammy. At this point, we will proceed to the, we would like to promote promote for more updates about the upcoming events of Philippine Planktology and also the collaborative events by Info. Please follow our Facebook pages in the Institute of Marine Fisheries and Oceanology and also Philippine Planktology. And also, we would like to emphasize and invite everyone, those who are interested to pursue their undergraduate or master's and PhD in fisheries. So the Institute of Marine Fisheries and Oceanology and the University of the Philippines Visayas has several degree programs, such as Bachelor of Science in Fisheries, Master of Science in Fisheries, Major in Fisheries Biology, Master of Science in Ocean Sciences, and also a PhD in Fisheries. So if you are interested or for any inquiries about these programs, you can head out to the Facebook page of the UPV Graduate Program Office, or you can email them at gpo.upvisayas at up.edu.ph. So next, we also have two upcoming lectures as part of the fourth Philippine Planktology lecture series. So the next speaker would be Dr. Patrick Pata. He will be discussing his presentation entitled Bridging Species Level and Community Level Processes Using Trait-Based Approaches in Zooplankton Ecology. And also on the August 12th, we have Dr. Le Chin Chu, who will discuss what can theropods tell us about evolution in the open ocean. So we will be posting the details of these upcoming lectures on on the Facebook page of Philippine Planktology, and also Planktonish Laboratory, and the info page. So please like and follow us so you can be updated with our upcoming events. And for our participants today, please wait for the link of the evaluation form. We will send that via the Zoom chat, so you can head out and answer that form so you can receive your e-certificate for this specific lecture. And we will see you all at the next lecture on August 7th, I mean. And with that, to end our program today, may I call in again Dr. Christy Acabado for her closing remarks.
Hello. I feel overexposed here today, but yes, thank you for coming, and I hope to see you again on August 7 at 9 a.m. Please watch out for the announcements on our Facebook pages. Thank you very much for always being with us. Bye. Sir Junie, we can end the Facebook
Thanks,
live.
everyone.
Okay,
Thanks,
thank you very
Tammy.
much. Thank you,
Thanks,
Tammy.
Sir Journey.
Maybe we can exchange emails later. Yes. All right. Thank you. Bye bye.